So, what do you do if you're pursuing IVF and you're offered genetic screening? Maybe it's PGT A, maybe it's PGT M, and now there's a new type of screening called PGT P. How do you know what they all mean and if you actually need them? Well, it's hard to know. But today, we're going to help you clear it all up by introducing you to somebody who knows all about this and is starting a company that specifically deals with an intricate type of testing called PGTP. His name is Dr. Jonathan Anomaly, and he's a professor, a researcher, a scientist, an economist, and so many other things. And he's going to tell us all about why these things matter and why there should be transparency so that you could understand all of the social, emotional, and scientific implications for all of these choices. From our Building Your Family community, this is the Building Your Family podcast. I'm your host, Lisa Schumann, author, researcher, and reproductive medicine therapist. Welcome back to a new episode of the Building Your Family podcast. I am so delighted to introduce you to somebody today who will give you so much interesting information that you haven't heard anywhere else. You know, very often we talk about genetic testing and what it's going to mean for you in your fertility journey. But today we have a special perspective from a person who is an author, a professor, and is a specialist in genetics. Dr. Jonathan Anomaly, and he's going to tell us all about all of the things that he's involved in, which you will learn about through the podcast, and ways to think about genetic testing and different ways of seeing things than you've ever thought about. Dr. Anomaly, I really appreciate you coming here today and sharing your time with us. I really like the audience to understand a little bit about you since you're not a reproductive endocrinologist and you're not a fertility specialist, but you're going to teach us all about genetics.
Well, I hope so. Thanks for having me. I won't bore you with too many details. My background as a professor is more in the social sciences. So I did something that's called PPE in the UK. That's philosophy, politics, and economics. And the kind of short version of that is economists look at the world through a kind of lens of how to model human behavior. Each person has their own preferences and they kind of sum up to an overall pattern of transactions. And philosophers think about how to evaluate behavior, especially in moral and political philosophy. And I got into this world some years ago because I was interested in the future of reproductive technology and what would happen in the big picture over 100 years, 1,000 years when people had the power, let's say, to edit their genes or to select embryos using what we're going to talk about today, polygenic scores, which is definitely not gene editing. But I'd thought about some of the consequences of these kinds of technologies and in particular, how they might go wrong. In the past, we had sort of the coercive eugenics movement where some people thought governments should either forcibly sterilize people or tell you who you should reproduce with, that sort of thing, but also how they could go right. We often hear about these worst case scenarios, about the kind of dark side of what could happen with reproductive technology. But I'm also interested in the bright side. I mean, there's a lot of good that we can do with this kind of technology. So that was my academic background in it. I wrote a little book called Creating Future People. I wrote some papers and then I got recruited to work with a company that does polygenic embryo screening. So I guess that's what we'll talk about.
That's so exciting. So exciting. So as I said, we're going to take a little bit of a different viewpoint here. Our audience knows a little bit about genetics from going through fertility treatment themselves, listening to people on our podcast talk about the pros and cons of using different sorts of genetic testing. And in our next episode with you, we're going to talk about how that is used in donor screening. But for today, let's talk about it with regard to people who have an opposite sex relationship and are trying to conceive on their own and trying to understand how genetic testing can help them or not, and if it's important for them or not in general. Perhaps we could just review a little bit about PGT M, PGT A, and then PGT P as you are the expert in that.
Sure. Yeah, that sounds good. So you've, I'm sure, talked quite a bit about M and A, but I'll just say really quickly what those are. So for about 30 years now, PGT A for aneuploidy has been available. And actually, one of the people who invented that, Santiago Munet, is on our team now. So that's kind of fun.
Oh,
He's completing the overall PGT spectrum from
wow.
A to P. But yeah, A is testing for chromosomal abnormalities, most of which are incompatible with life. So everyone has heard of Down syndrome, and people with Down syndrome can live good lives, as you know. You see people on the street. It's a disability, but it's a manageable one. But a lot of chromosomal abnormalities are much worse actually than that, and in fact will result in miscarriage or spontaneous abortion, as it's sometimes called. And so PGTA was and still is just a simple test to ensure that for a given embryo, it has the correct number of chromosomes. And when you do PGTA testing, it does raise the implantation rates naturally enough because your body has natural ways of detecting when an embryo has aneuploidy, and it often rejects it. So that's the so-called spontaneous abortion when you don't even know you were pregnant. It happens
Mm-hmm.
all the time. And so anyway, that was the first kind of genetic testing, aneuploidy. It's pretty routine in the US. Surprisingly, it's not as routine overseas. Maybe it's the cost. Maybe some people haven't caught up on the genetics. It's a little unclear, but that's A. And then M, which stands for monogenic. And that came out right around the same time. And that tests for single gene disorders of the kind we've all heard of. This is going to be boring, I guess, for doctors in your audience,
No.
but
Mm-hmm.
obviously things like cystic fibrosis or Tay-Sachs, hemochromatosis, and sickle cell anemia. And those all are in different regions. So sickle cell famously is in all of those areas where you find malaria. So it's sub-Saharan Africa, but also South India and parts of the Arab world, just because that's where the mosquitoes were that transmitted
Mm-hmm.
malaria. And as you have one copy of that gene, it gives you some immunity to malaria. But if you have two copies, you get sickled cells, which is really bad for you. So fast forward, we've all heard of the Human Genome Project, and you and I lived through that in the 90s and early 2000s. It took a really long time to get a first draft of the human genome, but that was published right around 2000. I remember when Bill Clinton announced it at a press conference with Craig Venter in San Diego. And even then, that was 2000, 2025, well, 26 years ago now. It's now 2026. It was just a draft. It wasn't even really the complete human genome. We didn't know about all the variation within that overall genome. That took a lot longer. And so fast forward now, took another 15 years or so to start building these so-called biobanks where, places like the UK, Iceland, Japan, now there's a lot of them around the world, they would ask citizens of their country, would you like to opt into this? What we'll do is we'll sequence your genome and, not sell it or anything, but sequence it and then ask you, do you have, particular conditions, and we'll match those against your medical records. And then what we can do is, if you sequence huge numbers of people, hundreds of thousands, and then see which ones have diabetes, which ones don't, which have Alzheimer's, which don't. So you need a diversity of age and sex and all these other things.
Mm-hmm.
Well, when you sum all of those together, you can do these genome-wide association studies and look for the significant genetic variants. And here we use the word genetic variant because, a gene is usually defined as something like a protein coding region of the genome. There are different definitions of it, but every A, C, G, and T in the genome is not a gene in that sense. It's a genetic variant where, it could be one or it could be the other at a particular locus. Anyway, when you think about, as we discovered after these genome-wide association studies, we look across the genome, you know, we share the vast majority of these genes and genetic variants, but there are sites at which we get these variations. It's a small part of the genome, but that small part is what explains why some people are more prone to type 1 diabetes or type 2 diabetes, why some people are more prone to schizophrenia or be taller than other people. And I use the term prone because, these are not guarantees of outcomes. These genes, we use the term polygenic. And yeah, I guess I should put this all together now. We went from PGT for aneuploidy, PGT for monogenic, and now through polygenic risk scores, which we use genome-wide associations to create. With polygenic risk scores, what we're doing is measuring the probability that if you implant this embryo will get diabetes or will have schizophrenia. But these are probabilities, right? The environment still matters. We're very clear about that. And we want to educate people. Nobody's guaranteeing outcomes. If you have a predisposition to type 2 diabetes, for example, famously, if you change your diet and you do, good exercise and you live a good lifestyle, actually, it's probably not going to be a big deal for you. It may or may not be, but you can really fix that. Type 1, less so, right? Type 1 diabetes is much more highly heritable and it involves your immune system. It's not just a kind of proneness to not be able to digest carbohydrates well. It's a bit different. So anyway, with all of these risk scores, we're just trying to quantify the likelihood that if you implant this embryo rather than that embryo, it's going to have a higher risk of diabetes or a lower risk of schizophrenia, et cetera. So I hope that helps.
I think for the people out there who are thinking about this, right, we can think, okay, we have an embryo and we don't want, as you said, natural selection to happen because that involves a miscarriage or that involves another IVF cycle or whatever it might be. And so, okay, that makes sense. And then I'm maybe prone to sickle cell or some other difficulty. So I'm going to ask to have this probe made for PGTM. So that makes sense to me. I can kind of check those boxes. Then why would you say to a patient, this is something else that you should consider? What are the benefits for them to think about?
Yeah, why would you do PGTP if you've already done A or M? So you've looked for chromosomal abnormalities, you've looked for single gene disorders. And by the way, most people don't do PGTM unless they have a family history,
Right.
a risk. So I'm Jewish and, Ashkenazi Jews famously have a high risk of Tay-Sachs. But actually, if it's not in your family, it's probably not something you're going to test for anyway. But yeah, anyway, so that's family history is often a reason to test for M, aneuploidy, especially over the age of 35. There are good reasons, but really I think it's worth doing for almost anyone. But for P, I mean, what's really interesting about polygenic traits and polygenic diseases is that they form the vast majority of diseases. So I like to use breast cancer as an example and ovarian cancer. So most people have heard by now of the BRCA genes, BRCA. It's kind of a
Yes.
hard word to pronounce because it's not really
Yes.
BRCA. It's like BRCA. Or,
It's missing a vowel.
Yeah, exactly. It's missing a vowel. But, there are actually 3 variants of that, which can raise the risk of breast and ovarian cancer. And my understanding is there's small differences between those 3, but my understanding is on average, if you have one of the BRCA variants, you have about a 50% roughly risk of breast cancer. It could be a little higher, and elevated risk of ovarian cancer. But what's interesting about that is, well, really twofold. One, it's not a guarantee you'll get breast cancer or ovarian cancer. And 2, only about 3% of all breast cancers are actually caused by the BRCA variants. So 97% of breast cancers happen either because of random environmental variables that we just don't understand yet, frankly,
Yes.
or because of polygenic predispositions, or both. And so it's a really interesting case because you think, okay, if I have this gene, this is really dangerous. And that's true. But you're missing the huge majority of even that trait. And when you move from a trait like breast cancer, ovarian cancer, to, let's say, schizophrenia or coronary artery disease, both of which we have good predictors for polygenic screening, those are always polygenic. As far as I know, in fact, I'm reasonably sure there are no cases of schizophrenia caused by one gene. And similarly for coronary artery disease, there might be some obscure one out there, but those are basically polygenic. And it's not just 3 genes or 10 genes. It's typically thousands and at the very least hundreds of these genetic variants. And so yeah, even with no family history of these diseases, it's often worth checking because anyone can have good or bad genetic luck. But I will say this, because I'm not a kind of salesman. I'm not telling everyone, oh, go get your polygenic testing. It can be costly. And if you have no family history of disease, which is unusual, but there are some people who basically low risk of almost everything, and you only have 2 or 3 embryos, well, it's probably not going to be as worth it compared with someone who either has a family history of, let's say, schizophrenia, coronary artery disease, et cetera, or who has a lot of embryos. Because the more embryos you have, the more genetic stratification there is, right? So the more you're going to see, like this one has only a 10% risk of breast cancer, and this one's say 40 or something like that. That's less, you're less likely to get that big of a stratification with only 2 embryos versus say 5.
Interesting. So let's say someone says, well, like for example, where I am in the New York area, for some reason that no one's able to pinpoint, in Long Island, there's an area where a large number of women have had breast cancer. And this is multiple generations. And nobody really knows. Is it because of the genetics of the people who've lived there? Is it something in the environment or the water? Who knows? But we hear things like this a lot. And so why would somebody then say, okay, well, if I don't have to worry about BRCA, if I don't see it in my family, maybe there are so many other variables in this problem that maybe it's not really worthwhile doing extra screening for.
I mean, I agree, actually. So I think until we know for any given disease or disorder, like let's say low functioning autism, for example, but you use breast cancer, I'll use that one as well. We're still not sure what's causing it. We know in both cases there's a genetic component, there's an environmental component, but we don't always know what that is. I mean, just to switch to autism and then back to breast cancer, it's like people are proposing all kinds of hypotheses for what's causing the uptick in autism. And actually, I think the most plausible one is just diagnosis. There probably is some environmental stuff that's raising rates a bit. There's probably some genetic component we don't understand yet. And there's overdiagnosis, or maybe it's just correct diagnosis and it was underdiagnosis before. But I think now with breast cancer, it's a little different than that. But similarly, I think there's almost certainly an environmental component. And I'm not an expert on breast cancer, so I don't want to speak to that. But I think the idea is, look, people do mammograms and as they should, especially at a certain age, or other kinds of testing. That all makes sense to me. But I mean, just to get into the sales pitch mode, I mean, I suppose if I were trying to sell you on polygenic screening, it would be, look, we all have polygenic risks. Literally every human alive has some probabilities of developing various diseases. And I would just say that, it's interesting information to know. It's not worth it for everyone, but I do think it's worth it for a lot of people. And I just say more embryos makes it worth it. Family history makes it worth it. But I will say, I've seen people where there's only 2 or 3 embryos, and even there with no family history, one of them may be at significantly higher risk of some disease. It could be a mental issue like bipolar disease, or it could be a physical one. And that's just because of the nature of how genes combine and recombine. So yeah, there can be reasons to do it for anyone, but I think the reasons are stronger when there's either a family history or, again, you have more than, say, 2 embryos.
So you brought up something that is, very controversial right now, but I'm going to go there anyway.
Okay.
We have seen a rise in the idea that there are certain emotional or psychological difficulties that should be all kind of categorized as disabilities across the board, and that there's something severely wrong with that. And there's been lots of talk, as I'm sure you've seen, about putting people in institutions for having all sorts of issues, right? And so I guess my next question is, if there's no natural or less natural selection, and I know this is your area of expertise, if there's no kind of natural selection, and I mean, I guess we can say maybe there isn't anyway, because certainly when I was a kid, there was no such thing as peanut allergies. Everybody ate peanut butter sandwiches in school. So maybe this is just our environment. If given that we don't have the same natural selection, are we then kind of siding with a lot of these groups that want to move into this? And I'm not saying we're headed back into a World War II situation, but are we headed back more into this belief that we should have one sort of population, that only a certain type of person should
Kind of
be
person should have
living?
children or something. Yeah. Yeah. I mean, I don't think so. So I always like to say, I mean, what really matters here, there are no guarantees against anything, first of all, right? I mean, dictators take over countries, cultures change in both good and bad ways. I don't have some magic recipe, because nobody does, right? To sort of say, well, my vision is always going to prevail because it's the right vision or something. So, of course, there are always risks to any technology, whether it's nuclear power, which can also be used to make nuclear weapons, right? Once you have that, that's why we don't want Iran, for example, to have nuclear power. They say that's all they want, but we know they want to develop nuclear weapons with that, right? And similarly, when you have the power to develop vaccines and do this kind of research, well, you might end up with COVID or something like this, when you're manipulating viruses. And I think the same is true for anything, whether you're, doing genetics in a really kind of unobtrusive, innocent way, you're just studying the genome. I suppose that knowledge could be misused by someone to do something bad. I think, look, in a society that prizes toleration and diversity, diversity in the deep sense of not just racial, but diversity of thought, diversity of, ways of living and things like this, mostly people are going to choose very different kinds of traits. And when you see the way that, for example, women who want a sperm donor choose a sperm donor, or men choose egg donors and that sort of thing, or couples choose those donors, they tend to choose all over the place. I mean, on the one hand, they want general health. That makes sense. But would you want someone who's more athletic, or who's a bit nerdier and smarter, or a bit more whatever, looks a little bit more like you? I mean, most people choose like their own race. Like you see an Asian couple will usually want an Asian donor and that sort of thing. And so I take it this is a natural extension of that. Generally, we all have preferences. I don't want to force this on anyone, but most people have preferences to have fewer diseases for obvious reasons.
Yes.
They want their kids to live long, healthy lives and be mentally stable and that sort of thing. But do you want your kid to be a musician or a nerd or a jock? I mean, people have different views on that. And I think the main goal should be to keep this relatively decentralized. I can imagine a role for government, like subsidizing genetic testing for people who can't afford it, especially if they have a family history of disease and so on. And you could imagine doing that to prevent massive inequalities between, let's say, the rich and well-connected and the poor or something, and access to this. However, I do see real dangers in governments getting really directly involved, because the more power governments have to make these kinds of decisions, the more incentive one party has over another party, to sort of use its power to try to manipulate these things. So that's my general answer. I think in a culture that's healthy and that prizes diversity of thinking and diversity of choice and so on, I think this technology is fantastic. In the wrong hands, yeah, I could imagine in a North Korea or something like that, it's not necessarily great.
Imagine your beautiful future. You're snuggling with your child. You feel so connected to them. Your friends and family are so happy, and you have everything that you've ever wanted. I want this for you too, but I want more than that. I want you to feel secure knowing that you've made the best decisions, not just short-term, but long-term for your future family. Wherever you are on your journey, whether you're choosing a donor, choosing a clinic, learning how to talk to your donor-conceived child about their origins, all of these decisions will have wider implications for your future. And I want you to have the right resources so you can feel empowered and confident at this very important time in your life. That's why I'm so excited to introduce you to Building Your Family. It's the only expert-guided platform designed for parents and parents-to-be using reproductive medicine. We provide unique strategies that you won't find anywhere else to make your path to parenthood smoother and more informed. And that's why our approach is about offering practical and actionable steps that you can use right now. Whether you're just starting out or already deep into your process, Building Your Family is here to guide you with expert advice, specialized education, stress reduction strategies, and a community to support you every step of the way. We can't wait to see you inside. So we will talk in the next episode about the difficulties in donor choice and this technology. But for a heterosexual couple who wants to have children who are genetically connected to them, because of course, if there was some significant difficulty, they might opt to use a donor, right? If they felt like, well, my partner really is all of these problems. Maybe I just want a donor because I don't want to take a chance. But more often than not, people would probably opt to say, okay, my partner has schizophrenia all over his family, and I want his genetics. And so this would be perhaps a good technology for me.
Absolutely.
Right. So in these situations, I can see how people would really want that. What are the other things that are available to them? What are other reasons why they would choose this technology?
Yeah, I mean, it's mainly disease risk, but there are aesthetic traits available. Like, so you can select on height, for example, and actually, it's height predictors are quite accurate. And there's even general cognitive ability, which is a bit taboo in the U.S., I guess, but it's not so taboo in most of the world. And what I've seen from people who do select on cognitive ability, they usually use it only in the balance, in the sense that they'll look at the traits of their various embryos and they prioritize health, especially mental health traits. And then, they don't want to be cognitively disadvantaged, for example. And so, if there's one embryo that's at, it's at reasonably low risk of disease, but it also happens to be, very low cognitive score or something like that, people will tend to select against it. And so that's some of the other traits that we can do. Some people think that's great. Some people that scares them a bit. But my view is cognitive ability is, for example, it's a health trait just like others. For example,
Hmm.
we know that intelligence, as measured by IQ tests, as flawed as that might be, they do measure something, and those things are correlated with a lot of different healthy life outcomes. So we know length of life, subjective well-being, and also things like intelligence is correlated with self-control and long-term planning, which in the context of medicine, it's hard for your listeners to imagine what it would be like to live life as someone with a 75 IQ or something like this, but it's a very different life. Not being
Hmm.
able to remember to keep your appointments, not being able to manage your bills and your money and things like that. There are a lot of reasons why being in that position would lead to less overall health and well-being. Now, just to emphasize, that's a reason we should take care of people who are in that position. We absolutely shouldn't, discriminate against them. We should have laws protecting them. We should always treat everyone in our society as moral and legal equals. Having said that, people will, for obvious reasons, there can be mental disadvantages and physical ones. And some parents want to see all of that information and kind of navigate accordingly. So.
I see. And are there other medical conditions that your service would allow for?
Polygenic screening? Yeah, I mean, one of the most interesting ones, and yeah, I work with a company called Harrisite. They're, as far as I can tell, the first to develop something called neurodevelopmental disorders, it is a kind of polygenic testing, but it's a bit different. So let's go back to sort of evolutionary theory in a way. When you think about an organism acquiring some random mutation, it could be from background radiation, it could be from an environmental pollutant, it can even happen during sexual recombination, right? There are new mutations that are introduced every generation. In evolutionary time, the vast majority of those would have been either neutral or harmful. It's very unlikely that any given mutation is going to be beneficial, right? And when you think about that, it kind of tells you there are many ways to damage the infrastructure of a genome, right? It's happening all the time. I
Yes.
mean, you probably have freckles and moles and things on your skin. I certainly
Hmm.
do. Those are kinds of damage. Maybe they're trivial, but some mutations, as you know, are quite damaging indeed. And there are very few ways in which you can actually improve something, so to speak, right? Whether it's a plant or a human or an animal. And so the point is, there are all kinds of mutations that occur, and some of those mutations happen in the sense that they control neurological development. And when you have some of those mutations, neurodevelopmental disorders, they correlate with things like some versions of low functioning autism, which is, as you mentioned, it's kind of a mess. What even is autism? It's a spectrum. It's all these things, but also things like dyslexia and other kind of minor disadvantages. But basically, these neurodevelopmental disorders can be captured by some of these rare variants if you whole genome sequence the embryo. And Actually, I should say something about that because this is another important thing that we've done, and I'm not responsible for it. I'm not smart enough to do this, but we have a kind of genetic superstar named Alex Young at UCLA, and we presented him with a fundamental puzzle. And here's the reason for the puzzle. In order to do really good polygenic prediction, you have to have a pretty complete picture of the embryo's genome. But it's very difficult to whole genome sequence the embryo. I can whole genome sequence you really easy. You can spit in a tube or a little bit of blood, and it's very easy to get a lot of stable DNA from those tests. And so you can do that for, $500. Really deep sequence, maybe 1,000. But embryos are very delicate. When you're biopsying the embryo to do PGT A, M, or P, you're just getting a few cells. So it's difficult, it's expensive, and very few labs can do it. So we can do that, but we prefer another method. And I'll tie this in with neurodevelopmental disorders in a second. The method that we came up with, knowing that every embryo is a function of parental DNA, just different shufflings, different combinations of the parents' genomes, we thought, you know what? What's interesting about the PGTA test that nobody is paying attention to is that it covers a very tiny fraction of the genome, and it indicates where sexual recombination happens along the genome. And so if we know more or less where the blocks of DNA, where they start and stop, where this one comes from mom, this comes from dad, this comes from mom, and we know just a tiny fraction of the genes within those blocks, well, we could fill in the huge gaps that remain from the PGTA test by deep sequencing the parents. And this is called short and long read sequencing, but I won't get into the details because maybe they're boring. But we realized if you could have the right algorithm, and it's a lot harder than it might seem. A lot of people have tried this until Alex Young succeeded at it about 1.5 years ago. If we could develop that algorithm, which we have, we could infer the whole embryo genome by simply asking people to ask for the raw genetic data from their PGTA test and then having the parents spit in a tube to recreate the whole genome. Now, putting that all together, that has two implications. One is we can actually look for, when we do these neurodevelopmental disorders, this test for them, as well as other polygenic disorders, we can actually look at the whole genome without whole genome sequencing the embryo directly. And if you have a kid who already has one of these, that can also be helpful. We can actually sequence the kid or sequence a relative who has one of these. But the second implication of this sort of inference of the genome from this common source of data, or it's called imputation, the second big implication is we can basically serve the whole world. All you need is a PGTA test. You thought you were getting a chromosomal abnormality test, but you were actually getting a schizophrenia test. You were actually getting a test for breast cancer, for Crohn's disease. And the reason I say you were actually getting it is you were getting it if we use that PGTA data and then sequence the parents. We do need to sequence them in the process to do that, but it really frees up the sort of patient to go to almost any clinic of their choice, as long as you have the right to request your data, which you do under HIPAA laws in the United States, and the physician respects your autonomy and your right to choose which embryo among the healthy embryos you want to try to implant. Then actually you can get polygenic testing and these neurodevelopmental disorder tests as well.
Wow, that's very interesting. But I guess then you'd have to have parents who are alive and available. And
that's true. In fact,
right.
if you don't have the parents, we can't do that unless we directly whole genome sequence the embryo. I will say one more thing, since you asked, and sorry to interrupt, but in terms of what else is coming, I mean, so we at Harrisite, we offer about 20, 25 disease traits and then some of these other non-disease traits, but there are a bunch more coming, and we have a list of probably 50 or 60 in the works, a lot of autoimmune disorders, et cetera. We don't offer everything we can predict because what we want to do is only offer traits, that is, disease traits especially, that we have validated using tens of thousands of sibling pairs. And the way that we validate is we want to make sure we do it across different racial groups, different ancestry groups. That's important because there's some small genetic differences there. But also, it's really important to do this properly. And I mentioned Alex Young, and he pioneered this method of, if you have a so-called polygenic score, you've developed it from a biobank, how do you know it works? That's the most important question. Not how did you develop it, but you're predicting
Yes.
future outcomes, right? So maybe you
Yes.
were going to ask
Yes.
that.
I was.
Yeah, good. So how do you know it works? And this is of crucial significance. And anyone who wants to get testing done should demand of the company, how did you validate your results, and how strong are your results? How powerful are your predictors? And again, we don't want to offer predictors until we've thoroughly validated them and they explain a significant fraction of the genetic variation in the population. So the way to do it properly is not to take a polygenic risk score and just run some random person's DNA through them to predict, say, their height or whether they have diabetes. I mean, it's an okay way of doing things, but it's imperfect. What you really want to do is take adult siblings, and we have data sets of tens of thousands of these siblings, and run their DNA through your predictors and see what you come up with. Now, the reason that adult siblings are important is pretty straightforward, and that is that adult siblings have the same genetic relationship to each other, which is they share about half their genes, and to their parents, which is they share about half of their genes with mom and half with dad,
Right.
as embryos do to each other and to their parents. And so what you really want to do is, if you want the most accurate scores, is test your polygenic predictors on adult siblings. And you need, again, racially diverse, age diverse. Let's say that you're predicting
Yes.
Alzheimer's, which we do. We have a pretty good predictor for that. There's one gene that really does raise the risk quite a bit. So there's partly a monogenic cause, and then the rest is highly polygenic. So the monogenic is APOE4, whatever. The name doesn't matter. They always have these technical, letters and numbers. But basically, yeah, you'd better have in your data set some older people if you're trying to predict, Alzheimer's disease.
Yes.
And so that's what we do. And if you have enough of these siblings, and you can run their DNA through your predictors and then see, what is their absolute score and their relative score relative to their parents, to the siblings, et cetera. That's going to be the gold standard for testing whether or not your scores work and your predictors work. And just to kind of cap that off, it is true there will always be an environmental effect. So one thing, please don't misinterpret me. I'm not saying diet doesn't matter, exercise doesn't matter, and even things like poverty, especially extreme poverty, maybe having a little less than average income, honestly, that doesn't really do anything to your DNA. But if you don't have the proper amount of basic nutrients, that's going to really impair your immune system, your brain, which is why,
Yes.
IQ tests, shouldn't be given to people who grew up in extreme poverty, because honestly, they don't really capture the full potential of what that person can do. So yeah, that's the idea, is you want to run these predictors through adult siblings, and then you get a pretty good estimate of how well your predictors work. And then you want to present to your customers, your clients, your patients, depending on, are you a company or a doctor, what's the uncertainty here too? Like, we're not giving you certainties here. What we're saying is, this is the best we can do. Here's how much of the genetic variation we can predict. There's still going to be some random chance and bad luck and environment, but all things considered, given these trade-offs, what do you guys want to do? What do the parents want to do?
Well, I think that's great. That's really helpful to know, because that was actually what I was going to ask after that. There's, such an interest, and we'll talk about this as we wind down into our next segment. But there's such an interest right now in the reproductive community about epigenetics, particularly because people really want to feel connected in any way they can think about. And so, since we have such a interest and so much more literature about it now, people are really focused on how can I maximize whatever I want to maximize. And, there's certainly been enough research to show us that it is significant. So if you're in a situation like schizophrenia, for example, you see a family where maybe an aunt has schizophrenia, and then maybe one of four children have it, and then nobody else has it. So why is that? Did the child have trauma? Was it environmental, where they lived? Is it the way they ate? Is it their birth order? What happened with that one child when there's just an aunt who has schizophrenia? So And of course, there's offshoots of that, right? Like bipolar disorder and different
Mhm.
ways that those sorts of symptoms can show themselves. But these difficulties have so many variables. Is it really even possible to say, we're going to test for schizophrenia?
Yeah, and I don't want to step out of line. I'm not a schizophrenia expert, but for any of these kinds of traits that are highly heritable, and schizophrenia is very highly heritable, one of the sources of evidence for why genetics matters so much is twin studies. So, twin studies are the kind of gold standard of the field of behavioral genetics. And that's the idea that
Hmm.
even before we knew anything about what genes are doing, I mean, say 50 years ago, I mean, yeah, the double helix had been uncovered, the structure of genes by Crick and Watson, but they didn't know anything about what particular genes were doing. That was all a mystery. And yet, even at the time, there were already some pretty good studies on how heritable particular traits are. And some are not very heritable and some are highly heritable. Schizophrenia is highly heritable. And the way that we knew that is simply to take these tens of thousands of twin pairs that were born and raised in very different environments. And they would take monozygotic twins, which share all of their genes, and dizygotic twins, which share half of their genes, and just watch the ones. They can consent them into these studies, follow them up throughout their life, and just see how tall are they? Do they have schizophrenia, et cetera? And you do need large samples, but after looking at tens of thousands of these twin pairs that were either raised together or in very different environments, you can thereby estimate, how much is a genetic contribution versus an environmental contribution. And
Hmm.
so we know, just in the case of schizophrenia, it's highly heritable. Nevertheless, that doesn't mean that if you have a predisposition for it, you're guaranteed to develop it. And my understanding is part of the reason could be what you mentioned. It could be partly epigenetic. It could be partly environmental luck, maybe some developmental factors in the womb that are poorly understood. Again, I'm not an expert. I don't claim to be, but my understanding is at least a fraction of it is just not understood.
Hmm.
Um, so basically, that's not to say that genetic testing doesn't matter. It matters a lot, and you can minimize risks through genetic testing, but you're never going to guarantee outcomes. And I want to actually put in one more word on that because we started by talking about breast cancer, and when people say things like polygenic prediction is merely probabilistic, what I want to know is this going to happen or not? And I like to say, hang on, guys, like that's not the standard of evidence. I mean, when you have BRCA1 or 2, it's a 50% risk, right? Nobody is saying you're going to get it or you aren't going to get it. And
Hmm.
what's the other 50%? And I think the answer is we don't know. There's some hunches. Maybe it's epigenetic, maybe it's random, maybe it's something that's just not understood yet.
Well, that's very helpful and such a great answer. And for everybody out there who's loving this conversation, please stay tuned for the next one. We're going to continue this in our next episode and talk also about what this means for genetic screening. We're going to talk about all of these different types of genetic screening again and make sure that you understand what this is going to mean if you're using a donor. So thank you so much for coming. I really appreciate hearing all you have to say, Dr. Anomaly. It's fascinating. And thank you all for coming. I really appreciate your time. And please make sure that you press subscribe, even if you're heading to the next episode, so we can keep going because this is how we continue to educate you. Thanks so much. Thank you for joining me today. I hope this was helpful to you. And if you'd like more personalized support, more information, and more education, you can find it in the show notes under the Building Your Family community. We'd love to see you inside. And if you were interested in today's topic, you will really like this video.